Okay, welcome everybody to the first of the afternoon workshops. Alexey Sergushichev. People butcher my name all the time. So he's going to be giving the package demo on easy access, interactive exploration, and analysis of public gene data sets with Phantasus. And I'll be moderating all online stuff. Questions.
I'm Alexey Sergushichev, which is like very hard to pronounce, especially if you need to like to call the bank and they ask like your surname. Okay, so I'll be talking about a package called Phantasus that's developed in my group. And before starting, let's go to this. So I have this environment prepared in workshop environment, the conductor. So let's go there and try to open it. Yes, it's not necessarily like you need to go with me, but you can do this. So there is a demo on this website, workshopbyconductor.org, and you can find it on the left side, like package demo Phantasus. You need to log in to this website with your login and password. I don't know if you have it. Do people have it?
If you don't have it on the screen.
Yeah, so if you don't have one, you can register there, and like it's free and pay. So let's run it, and while it's gonna load, so I'll talk with the slides. Okay. Yeah, so this is an overview of my talk. So first we'll talk about this Phantasus app that we developed, and like briefly with the slides, and then there will be 2 parts demo, and like more demo of the Phantasus application as a web tool, and a bit of demo of how to work with RNA-seq counts from Geo in our environment. And first, I want to talk a bit about why actually we started to create another gene expression analysis interface, as there are a bunch of other applications like this. And there were a couple of points that we were trying to target. Yes?
Because online they're still just seeing the Galaxy screen. We need to share it here.
Okay, I was told that just switching would work fine.
Is it possible they shared a window instead of the whole screen?
Yeah, but,
there's a screen.
there's a screen.
There's delay.
No, it's a browser, the same browser. So I can share menu. Let's try this. Share screen. Yeah, I have to share it on screen.
Yes, got it.
Okay. Yes. So there are a couple of points that we tried to pursue when we developed this. And first of all, it was visual analysis of the data, and like myself and like I believe that it's when you analyze the data, it's very important to have it visualized, because there can be a lot of artifacts there, and it helps to see the data with analysis. And in particular with, for example, gene expression data, especially like with public data that we work with, sometimes there are so many flexibilities in how you can analyze this, and like writing a script for a new data set can take a while if you have some kind of interactive application, it can be easier to do this. However, so there are a couple of tools that do like visual analysis, like heat map, and I will talk about one of them later a bit. However, as we know, like for gene expression analysis, most of the tools are in R and like in bioconductor, and we do want to be able to do this like proper analysis. We want to somehow interact with our environment. And also the last point is that there are a lot of public datasets in Geo, like as we also heard like from previous talk. And sometimes, especially like if you're like, I know I'm from Russia, and it's costs a bit to get your own data set and like to make it done. However, there are like a lot of publicly available data that you can leverage. And we wanted to have a really like, high coverage of being able to load datasets that are available in Geo and like, use your own dataset. And also, there are like I would like to talk a bit about what audience we target. And when we originally started to work with this, it was for educational purposes. Again, because like it's we wanted to have something visual, and for students who like don't know yet like how to program or like see for the first time generation data, like this visual like interface, I think could help. Also, like turns out that it actually helps people in like in real research. For example, if you have experimental people in your lab who don't know how to code yet, at least, but a lot of stuff they can like with tools like this they can do now. And later it turns out, well, not later, but at some point it turns out that actually this tool is helpful for bioinformaticians as well who can code. For example, I myself use this tool like a lot of time, even though I can do it actually because it's sometimes like for geodata, it's much faster to load and visualize it and process than to write the corresponding script. And with this, so like starting to like these features as this visual interface. So there is an application, like web application called Morpheus, which was developed by Joshua Gold at Broad, which was a kind of new generation of software, like an heir to like Genie software. I don't know if some of you used it a while ago, but we actually liked very to use some workshops on how to process data. And so in this web tool, it's like nice tool for the visualization of heat maps, which is a standard way to do to visualize gene expression data. It's like fast and runs in a browser. It doesn't send the data anywhere, which is also good. However, it's limited functionality because it was written in JavaScript. So any method that you want to use, it has to be available in JavaScript. Well, I think now there is like this web awesome implementation that maybe it's can circumvent this. However, at that time, it was like a really big limitation. However, it's also open source, and you can build on this tool, which we tried to do. And importantly, there is so now we have this web interface that we like, and the next point was to connect it to this R environment where all of the methods are. And for this, there is a framework. Well, now there are different ways to do it. Like most of you have heard of Shiny, which kind of gets you there, but actually for our purposes, framework called OpenCPU was very helpful. And basically it provides an API that you can use to call R-based methods and get the results in like a JSON format and to get them into your web application as well. And now when we have this OpenCPU server, which can interact with our JavaScript-based heatmap interface, so now we can use all of the methods that we love, like from dsig, like lim, whatever. And also there is a package called like GeoQuery, which can interact with GeoQuery Omnibus database to load the data in your tool, which gets us halfway into the like last point about high coverage geodata. However, there is a problem. Like if you work with gene expression omnibus before and with GeoQuery tool, that it works well with microarray datasets, which stores these gene expression matrices in Geo. However, well, nowadays people do RNA-seq mostly, and so like the current datasets are RNA-seq, and for some reason, gene expression omnibus doesn't store in any standard format gene expression matrices, and so we had to rely on some external projects like ArcGIS4 from Avi Mayen lab or D2 from Mark Zeman lab, who quantified a lot of the samples from Geo in a standard format, and now we can rely on this data to also to support in the Phantasus. And in the end, what we end up with is like this table that kind of dataset that we support, and you can see that there's like a lot of the microarrays, but also a lot of RNA-seq samples. There are, well, mostly they are human and mouse, and there are like about 100,000 datasets we call like fully support. That's basically if you open it and like it will fully open. However, it's sometimes hard to tell if it's like fully support or not, like based on just like a computational perspective. However, now like from datasets that you can open and get some data there, it's like around 140,000 datasets that are available that you can try to look at some data. And currently there are around like 100 and maybe 70,000 gene expression datasets in Geo in total. So it's like, I know, almost 80% coverage that we achieved, which is really great for us. And this is like one of the points that distinguishes from some other tools, which are, for example, works either with microarray or either with RNA-seq and so on. And so together, this made us allowed us to make this Phantasus. And if you don't know, like Phantasus is the brother of Morpheus. Phantasus application that integrate these whole things together and allows you to do like a lot of the analysis in this very intuitive and visual interactive way. And the paper came up just recently, so you can look there for more details. And however, like today we'll walk over some of the stuff that you can do with this. And this Phantasus application, there are like basically three major ways to work with it. One of them is just use official mirror that we set up at our lab. But also it's available as an R package. So it's also like open source, and you can install it from Bioconductor. And that's what we're gonna do like in the half an hour left. And also you can use like Docker image that we built and so on. And like, there is some documentation on how to install it properly. So I'll talk about some of these, but there are more detailed description there in documentation. And I think these slides will be available maybe somewhere, or I put them in the Slack, so the links are there. Okay, so this was like a kind of introduction to this application. So let's try to work with it. And for this, we're gonna use this like as a live demo. So I hopefully like everything's gonna work. And we're gonna try to set it up in this workshop environment and see how it works. Okay, so now I'm going to the browser. And so now I have this interactive tools running. So, yeah, and there is a vignette that we're gonna work with. Okay. And let's see. Okay, hopefully we can see this. Yeah, so there are like in the Phantasus package itself, there are like basically two major functions that we're gonna use. It's like setup Phantasus to set it up, so configure, and serve to start the server. So first, after like, we have this package, so we need to call. Actually, first we need to get all the libraries. Yeah, and this is like the first function, start Phantasus, which is kind of interactive. Will ask interactively to a couple of questions, like for example, where actually to put some of the files, but mostly by default you can use what it suggests. So we're going to use this cache directory to store some files. One of the things that is important is like some annotation databases that we're going to use, and it suggests to load them to the my lab website. But also it can rely on packages like org, hum, hs, gdb that are available from Bioconductor if they are installed. So they are not installed here, so it will download it. And these are like important for, actually we'll see for why it's important.
Does this get cached on the machine?
Yes, so like you need to do it like first time, and then we'll use it here. Yeah, and so we can download some. So this was annotations about like genes and gene relations like with different types of IDs. So now we want to download some pathways. Yes, let's download them. And actually that's it. And let's explore the actually configuration file a bit. Yeah. So this is the configuration file. So it's like pretty simple. Not much stuff there. So there are some of the things like host and port, where it tells the server like where to listen to which like port on the computer that when you run the server, which you can modify, but you can leave as is. Usually you don't need to modify this. Some of the things that we're going to talk later a bit, it is preloaded dir that allows you to put some data sets that you can pre-process there. And then there are a bunch of stuff that just tells where it's located. And the important part that we're going to talk later today is this one, which tells where from to get the account data. And this, so it can be also locally, so you can download some big files, but also you can work with some remote server that we have there. Okay, so this was config file, so you can modify it a bit, but we'll keep it as it is. And now let's try to start the server. So the server, so you can actually you can start it within a studio, but it's a bit easier to start it in like a new process. So this like a new terminal. So this tells you the command that you need to run to start the server, and yeah, and we specified this preloaded directory because we didn't modify the configuration file, so we just created empty one, and we're also going to use it later. And so you can either run it in the terminal manually, but also if you run the next line, it will communicate with RStudio and and we're going to start it, the application in this terminal window, the second one. Okay, and it's like says some things, but most importantly, yeah, so it said that like server was started at this host and port. So now we can load it, and for this you're going to use this browse URL function. Basically, so we list the servers listen to this local host at port 8000. So let's go there, and RStudio will open this this URL in the new tab, and this is like the home screen for this application. So there are a bunch of datasets there, like from TCGA, but what we're gonna be interested in is datasets from Geo today, and yeah, and today I'll talk a bit about a couple of datasets. The first one will be this microarray dataset. Oh, it's kind of microarray. I know people are familiar with now, but it's like a good example of the data. And actually, there are still a lot of helpful microarrays in the geo. Like half of them are microarrays, half the dataset are microarrays, and this is like just a good example of the dataset and how you can work with this. But also, like the next one will be RNA-seq. And for this, I'm gonna use this basically try to go over this quick start tutorial that is on the link here, and also the link is provided in the Markdown itself. Okay. Yep. So so in this menu, it's like we can load different datasets. So one of them also we can load some datasets from our own computer, from core facility if you have this, or if you store this. But we're gonna use these geo datasets, so we can enter some GeoCID, like dataset ID from geo, load the dataset, and in the meanwhile, if you look at the server, like terminal window here, it's download something from geo, and also like it's caches there. So like next time if you're gonna open this dataset, it will be open faster. Yeah, and it worked. So basically here we have this heat map that represents our dataset. So we have the bunch of like 16 samples, which are represented by columns. We have a bunch of rows, which are microarray probes that correspond to genes, and we have a like standard heat map, row normalized from minimum to maximum value, which is represented as colors from blue to red. And so immediately, like with this macro dataset, you can see that this data, like, and when I point, it shows the actual values there. So we can easily see that it's not log scale, there are big numbers. So, and for analysis, we need to normalize this data and scale it to logarithmic scale. So you can easily do this as well. We can go adjust, we apply log scale normalization, and also we're going to apply content normalization at the same time. So we click OK. And a new tab appears because we modified our matrix. And each time the gene expression matrix is modified, the new tab appears so that you can go back if you did something, if you want to go back. So now we can also like observe this. Yeah, so now we have this log scale data. And yeah, so one of the problems is microarrays. Well, not problem, but a feature of microarrays is that there are multiple probes per gene. And for example, if here's a search button, so I can check my favorite gene, well, one of the favorite genes, actin B, which is a like housekeeping gene that people use as a control, which should not be changed, for example, and like on qPCR analysis, for example. Yeah. And we see that there are like 5 probes that correspond to the same gene, and there are different ways to deal with them, but usually it's easier to work with gene level data as opposed to probe level data. And so we can do this collapse tool. Oops. So we can select the unique gene IDs, so that corresponds, that has the maximum average expression. So we just select the probe that is highest average expression. And so now we have this 20,000 genes as opposed to 40,000 probes. We can calculate. Next, we want to filter. We can filter, for example, filter out lowly expressed genes. So we can create this calculated notation with operation mean, which will create a new column here with average expression. So now you can, for example, sort by it and see that well, there is a bunch of genes, like highly expressed genes, which is some pattern of expression, and like lowly expressed genes with a lot of noise. So we can, for example, filter these genes out, like the lowly expressed ones. So we're gonna do this filter step. So we can use this mean column as a filter, and here we can either put some values if we do know some particular thresholds, which or like what I usually do, again, is just to put numbers like around. So I prefer this number 12,000 or just 10,000, something like amount of expressed genes in a single experiment. And I'm gonna create a new heat map. So now we have like the minimal value of expression being 5.5. So here we have kind of normalized in preprocessed our data set. So the second, another, so what we're gonna do. Yeah, so let's a bit explore this data set a bit. So typical exploration method is like PCA, for principal component analysis. So we can do this here as well. And I think this is one of the important methods that rely on R. So in the meantime, so it's accessed, actually, can check that, can I check? No, it doesn't show it here, but for these operations, so it goes to R and asks like, can you do the PCA for this data set that is stored on the server side? And this is like plotly.js plot that we can use like to color stuff and so on. So here, for example, we can see that, well, there is some, in general, the data set is good, but there is maybe something weird going on with these first replicates, which are a bit kind of diverge from the group. And we can also see this, for example, another way, if we do, let's do this, for example, clustering of the samples, right? So we can see that these groups are clustered together, and these first samples are a bit of clustered separately. And also, like what I really like to do is k-means clustering of the genes. So now we have these clusters on the gene side, so we can sort them and do this what we call like bird's eye view of the data set, which I think like very helpful to get your idea how your data set look like. So again, like on this, so now the genes are clustered together by their basically by their patterns of expression. And now we can easily see that, well, there indeed something wrong. Well, maybe not completely wrong, but something a bit different going with this first replicates in each of the groups. And they're not clustered here, and there is some different patterns of expression. And so what we can do, well, in this case, for example, we can decide that, well, these guys are outliers, so maybe let's remove them for the proper analysis, for the downstream analysis. So we can just unselect the samples and create new heat map without them. See that like it's a beautiful data set, like everything is clustered together, and we can analyze it further. Yeah, and now we have the data set cleaned up. We can do easily again differential expression. So we support Lima pipeline, in particular for microarrays. So we can select, for example, this interferon gamma plus LPS treated samples as a target and untreated samples as the reference. And we have these results of the differential expression appear near our, like with our, as a column counts in our data sets. So here we have like T statistic, P value, and so on. So we can, for example, sort by T statistic to find what are the most upregulated genes that are there in the data set. And yeah, and also like now, given that we have differential expression analysis, which is kind of one result of the normal analysis. So it's for some people, you can just look at the genes and see like what's going on. But also you can do, for example, path analysis on this. So you can do this again like with a bunch of web tools that are available there. You can just like, for example, copy this gene there, like in WebGestalt tool, for example, or whatever. There are a couple of things that we have embedded in is like ISEnricher website. Actually, let's maybe this. So we can select this top genes. Yeah, from the like top regulated genes. Well, we can also do it by filtering, for example, and we can do path analysis with Enricher. And Enricher needs symbols, so we select gene symbol column as our input, and we click submit. Yeah, and basically it submits these genes to Enricher website and produces this tables with the class analysis website, so we can see some transcription factors, for example, that are expressed, like regulated, maybe potentially regulated here, or like pathways, ontologies, and so on. Or we can do then analysis with FGSA. Actually, let me do this in the next slide. So basically, this kind of analysis, I think, well, it took me, I don't know, 10 minutes maybe to do it here, explain what's going on. But actually, like if you do it like a couple of times, it takes, I don't know, 1 minute, like 2 minutes to do this analysis. And actually, for me, even I do know how to process it in R. It actually will take a lot of time, especially given that we want to like remove some outliers, so maybe like re-annotate the samples and so on. So actually, if you want just to quickly glance over the dataset, I think this is a very helpful tool. And so this was microarray, and I talked about, I think, most of the stuff here. Yeah, so we're going to do FGSA later. And also there is, as I told you, that we support RNA-seq datasets. So let's open this dataset, GSC 53053, and we can use this link. So basically, we can use this GeoID to pass it as a part of the URL. So let's run this one. Yeah, it's also, I guess, download something from Geo, and also like it loads some data from our server that stored this gene counts. So now we have this data, and like this is RNA-seq, so this is RNA-seq count. So now we can see that numbers are here again, like weak, basically read counts. So yeah, so for RNA-seq, well, we do support DSeq. So actually, you can use DSeq tool for running the expression here. And actually, let me do this. Yeah, it takes a bit more compared to Rima, because we also use this log-fold change shrinkage there. Oops. Which I didn't install. Yeah, that was unexpected. I didn't try this. Yeah, but like in the function we use this APGLM log shrinkage, which I need to provide the dependencies. Yes.
I'm wondering if you went back to the RStudio and then you installed GLM, would this automatically work? Or you have to restart it?
It will automatically work. So it uses the same R environment. Look, I need to restart the server.
You would have to restart the server.
Yeah, but it will automatically. And also, the good thing is it stores. I think I'm not actually. That's a good question about this when you do it from R package, but actually you can specify. So this OpenCPU. So it has these sessions. So basically, when you do a call to the function, it creates like a new session which stores all of the data, and basically this data is stored on disk. So you can, for example, if you specify this location for OpenCPU session storage, you can like close this application, close the server, and restart it, and all of the sessions will be there, so you just can continue to work with. But yeah, let's not gonna do this right now. So another way, but with DSeq it's a bit like weird that it's kind of in this setting that it's easier to visualize log scale data, and DSeq works with raw count, so kind of you need to first do digital expression and then some kind of log scale transformation to get there. But also you can do it like with Lima pipeline, with Lima VUM pipeline. So we can use this VUM normalization, which needs, which is better to run with design matrix there. So we can specify the factors that are there in the design before running it. And in this case, it's just like simple design with 3 conditions. So we can use this factor condition like the only factor, and we can show the design matrix, but it doesn't matter now. And we can also like automatically filter lowly expressed genes with some thresholds from like function from Azure, I think. And click OK. So inside, it's gonna do like normally just log CPM, like columns per million normalization, but also, so this is the values here. So these are log scale, log transform data, but also it's within inside internally. It has the some so-called precision weights that are going to be used when we next do differential expression. Yeah, but again, like for this log scale data, we can now, and yeah, you see that now we have 15,000 genes instead of these 50,000 genes that we have from gene code annotation, I guess.
We have a question from the chat. Is it okay to ask them?
Yes, sure.
Yuri asked, how about reproducibility? Can you see what you did at each step and what parameters have been used? And can you generate an R-flow to
yeah, that's a good question. So currently, like we at some point, so because this is all mostly R functions going underfoot, so you can try to generate the code to do the same analysis. Currently, we don't support it. At some point, we try to do it, but there are some technical problems with that. So currently, we don't support it, but yeah, maybe we need to implement this. Yeah. Okay, so this is dataset. So what happened? Yeah. Yeah. So again, we can do like a PCA plot. This dataset is kind of okayish, and do some differential expression. Yeah, so now it's going to, because we use limo pipeline currently, so we're going to use limo pipeline, and also we're going to use this advanced design function that actually uses this design matrix internally. And here we have target level and reference level, and also you can have multi-factor design as well. So for example, if you have like patient data, like this paired like donors, we can use this multi-factor design as well. So click OK. And again, we have our differential expression data. Now, if you're going to want to use path analysis, for example, for FGSA, which we going to use right now, for this, we need like pathways are normally stored as trace IDs internally. And we don't have trace IDs here, so we have ensemble IDs and gene symbols. So we're going to use this annotation tool. So we can annotate from database that we downloaded before. So we can just convert this ensemble column from ensemble type of IDs to trace IDs. Now the problem is that not every ensemble gene has like a trace ID, so there'll be some NAs or some duplicate genes. So we are again going to use this collapse tool to have only one gene, like only one trace gene in the table. Yeah. And so now we can actually do this path analysis with FGSA, for example. We can select some database. Here we have, for example, hallmarks. I'm going to use this T statistic to sort the data, and here's our trace IDs column, which would be the same as in the database. Okay, so and here we have our path analysis results that we can look at. For example, we can check some of the genes here, and oops. So we can copy these genes from the pathways and use this to look how these genes actually look like in the whole data set. So again, let's sort our data and use this fit to window and see that, well, like each line here corresponds to the gene from the pathway. So you can see that indeed there is some nice enrichment of these genes in this differential results. And we can use this GSA plot function to not by average expression, but by like t-statistic, and we can see this nice enrichment plot with like, very significant p-value. Yeah, so basically we can analyze RNA-seq as well, and actually also it took me some time to say what I'm doing, but actually if you know what I do, it just takes very small amount of time. Yeah, and also, yeah, so I think to finish. So one of the things, so this stuff you can do with the server that we already deployed. So one of the particular features that is helpful to maybe deploy at your own like department or whatever is to have this preloaded dataset. So for example, you can, if you have processed some dataset for your collaborator, like as I did here, we can save this dataset as an R object into this folder that is created before, and now we can load this dataset by this link that we can share with collaborators and to be pre-processed with whatever data you have. Okay, yeah, so it's helpful to share with collaborators, or maybe even in publication. So we use it, for example, in publication. And can I get there? No, I can't. Okay, yeah, and just like final bit, like one last minute. So basically, I talked about this web application as a kind of user to visually work with the dataset, but also I think there's a couple of like some helpful things that we developed in the process. Or this like this is Phantasus slide package. You just have much fewer dependencies, and a particular function that is useful is this load counts from HSDS. And HSDS is like this remote, basically file system or server for working with matrix data. And we have this server setup that stores this matrices, this gene expression from ArchS4 or D2. And let's go on. So this is the second part. So yeah, so if you open this dataset GSC53 or 53 in R, like from using GeoQuery, and look at the expressions, you're going to see the empty matrix because there is no data in Geo for this. However, now let's skip this one. Let's skip this one. Yeah. So, but using this function load counts from HSDS and providing this URL where with our server, for example, you can. Oops, I need to. Yeah, so it basically asks the server, like, give me this count for these particular samples, and now we have this counts data there as well. And for this we used like ArchS4 database, and also have this gene annotations there as well. So like it's helpful if you need to process some public datasets in R. Okay, yeah, let me summarize this. So first of all, this Phantasus application, I think like it enables like for a lot of people to work with public data and like their own data, and it's helpful like not only for people who can't or like don't know how to code, but it's actually for bioinformaticians. I think it's also like to have a quick glance on the dataset really helps. Also, like, there are some particular functions that are helpful for if you to set up it on your own if you need, for example, to share these data sets. And you also can share not only data, but also how it's visualized, like the color schemes, clustering, and so on. And also this, if you need to load the data from Geo, like RNA-seq data, you can use this Phantasus slide package, and you can read more in this paper. And finally, I want to acknowledge the developers of this tool, which is mostly Maxim Klerov now, and also Daria and Vladislav, and some other people. Okay, yeah. now I'm finished.
Okay. Thank you. You could have asked them at any time? No. This looks pretty useful. I do think the adding in that it would spit out the code that it did would be very useful.
Yeah.
Okay, well, there is a snack break, but before we are allowed to get food, we are wanting to take a whole group picture, and it's gonna be right out there, right by the food, so that'll be really tempting, but but we're supposed to corral everybody to get a picture first. All right. Thank you very much, Anna.